Hello, I'm Ayse Dincer. I'm doing my PhD in computer science at University of Washington. Today I'm going to talk about my project, Adversarial Deconfounding Autoencoder, for learning robust gene expression embeddings. Gene expression profiles provide a snapshot of cellular activity, which allows us to understand the biology of various diseases. And I believe that unsupervised deep learning has enormous potential to extract important biological signals from expression profiles. First of all, because it gives us the ability to train without supervision, which is quite useful because it is very difficult to get a high number of samples with coherent labels. And also, they allow us to extract patterns without any restrictions or imposed directions, which might make it possible to learn something new. Unfortunately, it is not straightforward to employ these promising models on gene expression data because they often contain out-of-interest sources of variation in addition to the signals we seek. For instance, we can observe variations as a result of out-of-interest biological variables such as age or technical artifacts such as batch effects. We call these biological or non-biological artifacts that systematically affect expression values confounders. To demonstrate how confounder signals might dominate true signals in gene expression data, here we have a breast cancer expression dataset from two different studies. And we first plotted the top two principal components of this expression matrix and observed that the dataset effect seems to be encoded as the strongest source of variation. We then simply passed this expression matrix to an autoencoder and created the same plot for the autoencoder embedding. And again, we observed that the dataset effect is highly confounding the space. And this problem becomes even more severe when we try to transfer our models to unseen datasets. We address this problem of entanglement of confounders and true biological signals to show the power of deep unsupervised models. And our aim is to learn deconfounded expression embeddings. To achieve this, we propose Adversarial Deconfounding Autoencoder, ADAE in short. ADAE is an extension of autoencoders and it consists of two networks. The first one is an autoencoder that is optimized to generate an embedding that can reconstruct the original input. And the second one is an adversarial network that takes the embedding generated by this autoencoder as input and tries to predict the confounder. And our goal is to learn an embedding that can encode as much information as possible while not encoding any confounding signal. And to achieve this, we train these two models simultaneously. The autoencoder tries to reconstruct the data while also preventing the adversary from accurately predicting the confounder. But at the same time, the adversarial predictor still tries to improve its performance. So the two models are competing against each other until they learn an optimal deconfounded embedding. We tried to show the broad applicability of our model by exploring different datasets and different cases of confounders. And we evaluated our embedding in terms of how well it can deconfound the space, preserve biological signals, and transfer across different confounder domains. We showed that ADAE can learn deconfounded, yet biologically meaningful embeddings by training a standard autoencoder and ADAE where dataset is the confounder, and observed that these clearly separated datasets in the autoencoder embedding space becomes fused and indistinguishable in the deconfounded space. But more interestingly, when we color these embeddings by different informative biological phenotypes, we see that ADAE can clearly distinguish the samples by these phenotypes, which is not possible for the autoencoder embedding. We also investigated how successfully ADAE can generalize to different domains by transferring the models to an external dataset unseen by the model. And when we compared the embeddings learned for the training samples to the external samples, we observed that ADAE can manage to map the external samples to the same manifold as training samples, which is totally not possible for a standard autoencoder. Furthermore, we quantitatively measured the performance of ADAE by comparing it to alternative deconfounding approaches. And to do that, we fit prediction models on the embeddings and predict various phenotypes of interest. These experiments allowed us to demonstrate the superior performance of ADAE. We also experimented in different datasets and confounder settings. For instance, we used sex labels as the confounder and transferred the embeddings across confounder domains, such as training on males and predicting for females. Another cool feature of ADAE is that it can work with continuous valued confounders as well, for instance age, which again allowed us to carry very interesting embedding transfer experiments. To summarize, we introduced Adversarial Deconfounding Autoencoder to generate expression embeddings that are robust to confounders, can preserve biological signals, and transfer across domains. We're looking forward to extending our model by exploring new datasets and confounder settings. Thanks for visiting my poster.